Hello, everyone, and welcome to Chapter 12. This will be part one. We're going to go kind of on the front end. First half, we'll kind of focus more on structure, parts of neurons, types of neurons, things like that. Second half is when we're really going to focus on the physiology of neurons, where we actually talk about action potentials and all that good stuff. So let's get started. I chose the quote for this one. I know it's not what the comedian Stephen Wright intended, but I'm using it as a motivational quote, is that hard work has a future payoff. Laziness pays off now. So I know he's on the short term, laziness pays off because you get to be lazy, but laziness pays off now by bad grades. And that's what I'm going to say. So let's move on and let's take a look at this. So this is what we're going to be looking for. Remember, these chapter objectives, I always test on every one of them, so kind of treat them like a checklist, so to speak. That's just the way I write my exam, so I make sure every one of those are on there. So anyway, let's get started. Let's really get started on this and just basically understand what our nervous system is. And really, if we just want to be very big about it, big picture science kind of deal, is this is all the neural tissue through the body and all of your neural tissue. That's your nervous system. Now, neural tissue, remember, we talked a little bit about this when we did the histology section, and we said there are two kinds of cells here, and I called it Batman and Robin. Now, when I called it Batman and Robin, or Batman and Alfred, one of the ways we might think about it. Batman and his supporters, okay? We have the neuron, and that's Batman, performs all the communication, the information processing, the control functions, all the nervous system stuff. It does the main work. Where neuroglia, sometimes called glial cells, they are supporters. They're like Robin and Alfred. They help Batman, okay? Now, when we also look at this, we can see that these tissues, these two cells, create the nervous tissue, the neural tissue that creates the nervous system. And these cells, they help create the brain, the spinal cord, the sensory receptors, and all the nerves that link them together. So we're going to see they really are a very important thing. Now, just real quick, I'll talk a little bit more about this as we go along. We'll basically see more information about this. But we want to really take our nervous system and divide it functionally and anatomically. So let's start off anatomically. Anatomically, there is a central nervous system and a peripheral nervous system. Now, our central nervous system, that's our brain and spinal cord anatomically. So that's in the center of the body. It's found in the center, in the skull, in the vertebral column. So we have a central nervous system. Peripheral nervous system is everything on the periphery, on the outside edges. It's everything else. All neural tissue outside CNS is peripheral. So CNS, brain and spinal cord, everything else peripheral. Okay? Now, there is a sensory and motor aspect to our nervous system functionally. In anatomy circles, we call this afferent and efferent. Afferent arrives. So what arrives is going to be sensory information. That's what arrives at the CNS. Efferent exits. What exits the nervous system, the CNS? That would be motor commands. So afferent is sensory, efferent is motor. Okay? Now, the CNS, what it's got is your brain and your spinal cord, okay? Your brain and your spinal cord and the CNS. Now, what does the CNS do? Why do we have it? Well, what's going on, guys, is this is going to be our integration center. This is going to be the center that takes all the information in, processes it, makes sense of it, so to speak, and sends out the right motor commands. So, for example, if we are looking on the other end of a crowded room and we see a very attractive person and we get up and we walk over to them and we ask them out, well, what we've done is use our CNS to integrate that information. The sensory information was brought in. We had to first realize they are attractive and I want to go over and talk to them. Then we have to make the motor command to go over and talk to them and get their information about them. Say, oh, what's your name? What do you like to do? You want to go out with me? You want to go have coffee? Whatever. Okay. So we use this CNS to integrate information, to process information, to understand the sensory information, and make appropriate motor commands. Now, your brain, that's your higher order functions. And what do we mean by higher order functions? We're talking about things like your intellect, your memories, your emotions, things that basically make humans humans. Sometimes I say you don't have a nervous system, you are a nervous system. One of the ways I think about it sometimes. So, all right. Now, let's take our peripheral nervous system. Now, our peripheral nervous system, this is everything else. This is not brain, not spinal cord, everything else. Okay? Now, this is all the neural tissue outside the CNS. This carries information to the CNS, afferent. It carries motor commands away from CNS, efferent. There are bundles of axons here. And what we call these is nerve fibers, these axons. And if we bundle these axons, nerve fibers that carry it, we call them nerves. Nerves are made up of bundles of axons. We'll talk about that in the spinal cord, spinal nerve section. Now, there are two types of nerves in the PNS. There are cranial nerves, which those are on the brain. There are 12 of them. And there are spinal nerves, and they're on the spinal cord, and there are 31 pairs of those. So 12 pairs of cranial nerves. So that means there's one on each side, one pair on each side for both of these. Okay, cranial nerves, there's 12 pairs. Spinal nerves, there's 31 pairs. Kind of easy to kind of get those numbers into your head after a while. All right. Now, functionally, the PNS is afferent, efferent. Now, just kind of a reminder, afferent arrives. That's CNS, arrives to the CNS. Okay, that's where it's arriving to. So that means it's carrying, it's part of the PNS that brings sensory information from receptors to CNS. It arrives. Efferent exits CNS. This is carrying motor commands from CNS out the PNS, and it goes to what is called an effector. An effector is any cell that can respond. For example, a skeletal muscle cell at the neuromuscular junction. When we have a neuromuscular junction, we have that. We have a gland cell, neuroglandular junction. We can have nerve to muscle fiber, nerve to skeletal, cardiac muscle, nerve to smooth muscle, all those kinds of nerve to muscle, neuromuscular junctions. Okay. Now, the cell that can respond is called an effector. Okay. All right. Now, efferent can be further divided. Efferent can be divided into two motor systems that are distinctively different. There's a somatic and an autonomic. Somatic nervous system is all about skeletal muscle. Kind of easy to remember. Somatic, your body, somatic, is made up of skeletal muscles. So they control your skeletal muscles. Now, this is voluntary control, but has reflexes involved too. But the voluntary control of skeletal muscle is going to be somatic with reflexes of skeletal muscle as well. There's autonomic nervous system. We call this visceral motor. Now, very important. It controls smooth muscle, cardiac muscle, glands, and adipose. Okay. Now, I always put adipose here. Adipose is going to be unique to sympathetic, but smooth muscle, cardiac muscle, glands, and adipose. Now, they are involuntary. So this is the stuff you don't control. It's unconscious. It operates without you thinking about it. What I always like to say is autonomic nervous system allows you to do what you need to do without thinking about doing it. Things like breathing and your heart beating and your glands secreting and your kidneys filtering and doing all this stuff and your gut digesting. All the things that your body needs to do that you never have to think about is all done by your autonomic nervous system. Now, autonomic nervous system has these two really fascinating kind of things that go on in it. And what's fascinating about it is they're opposite and opposing. So what we mean is these guys are opposite in every way. And then in A&P, I will talk about A&P2 specifically. I will be talking a lot about, in the first chapter, how these guys are so different. We'll talk about how they're anatomically different. We'll talk about how they're physiologically different. They are the opposite to each other. They are the antagonistic side. They are opposing. Okay. Now, sympathetic, I like to call fight or flight. Now, I'm just going to introduce it right now. We will learn about this in detail when we get to the autonomic nervous system, but it's fight or flight. Basically, when I say fight or flight, it kicks in during stress. It's one of the things I like to say about it. Kicks in during stress. Now, it gets your heart rate up, gets your breathing rate up, but it slows down your digestive processes, for example. Parasympathetic, I like to call rest and digest. It kicks in during peace. It slows down your heart rate and breathing rate, but it gets digestion and go up. It kicks in during peace. Okay? So, if we look, we have anatomically a central nervous system and a peripheral nervous system. Central nervous system is made of brain and spinal cord. Peripheral nervous system is made of cranial nerves and spinal nerves. anatomically. Functionally, we have an afferent division and efferent division of our peripheral nervous system. Our afferent division arrives with sensory information to the CNS. Afferent arrives. Efferent exits. That's motor commands. There's somatic nervous system controls skeletal muscle, and there's autonomic nervous system that controls smooth muscle, cardiac muscle, glands, and adipose. There's parasympathetic and sympathetic nervous systems involved here. Okay. Now, sympathetic is going to handle the adipose. Okay. Now, the cell that does it all, the big kahuna, the important, the Batman, the hero of our story, is our neuron. Neuron is the cell that does all the communications. Now, if we look at it, it's going to have a large part, the big part of it, with kind of bulky anyway, is called the cell body, sometimes called a soma. Now, it's going to have most of your cytoplasm and hold the nucleus in it. So the nucleus is there, most of the cytoplasm is in there. Off this, in yellow here, there's going to be these branches that come off. And I like to call them the ears of the neuron. They are the branches that communicate with other neurons. They receive the signals. They're going to be like the ears of the neuron. The axon in this kind of red color is going to be the long, thin process that branches off and sends a signal. It's going to be a long, thin process that comes off that conducts what we call action potentials on a neuron. Then at the end, there are distal branches, at the distal end, distal branches, called telodendria. And on the end of them, if you look at the very end, you will see these little weird little bulges right here. Here, 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 here. These are called synaptic terminals. And we talk about synaptic terminals when we did neuromuscular junction. Okay? Now, let's start with the neuron cell body. If we look at a neuron cell body and we look in it, we're going to see cytoplasm. That cytoplasm is called the perikaryon. Karyon is a word, you've heard that part of a word before. You've probably heard the word prokaryote, eukaryote, things like that, karyotype. But prokaryote, eukaryote, those are really big words used in biology classes. Karyon references nucleus. It actually means kernel. So perikaryon means around the nucleus. What is around the nucleus is going to be our cytoplasm. So the cytoplasm in the cell body is called perikaryon. Now there is a structure inside the neuron cell body called the Nissl bodies. These are the rough endoplasmic reticulum. That's going to be where a lot of your neurotransmitters are made. A lot of your neurotransmitters get synthesized here, especially those that are peptide based. They're going to be made here. So I'm going to say most neurotransmitters are at least involved in their production are here. This is what gives gray matter its gray color. Now, if we think about it, most neurons are going to lack a centrosome. They're kind of stuck in that G0 phase, and they cannot replace themselves when they're damaged. Once they're destroyed, they're gone for good. We can't get them back. Kind of a big deal. Now, axons. If we look at the axon, if we look inside the axon, we're going to see it has a cytoplasm. Now, just like how we had the perikaryon in the cell body, the soma, we have axoplasm in the axon. Now, like how we had the sarcolemma on a muscle fiber, we have the cell membrane of an axon. It's called the axolemma. Where the axolemma is thick and attached to the cell body, it's called the axon hillock. After the axon hillock, there is an initial segment. Then we go to the axon proper. At the end of the axon are telodendria, and they become synaptic terminals. Synaptic terminals hold neurotransmitters. We sometimes call these axon terminals. So right here we have our neuron cell body with our perikaryon fluid in here around our nucleus, Nissl bodies in here. Okay, on here are dendrites, our axon hillock, our initial segment, and our axon proper, our telodendria with synaptic terminals or axon terminals at the end, going to the dendrites of another neuron, making dendrites like the ears of the neuron. Okay. The axonema is the axon's membrane. So axoplasm is inside the axon. Now let's talk about a synapse. Now, we did the model synapse. We did the neuromuscular junction. That is the model synapse. That is the synapse. If you can understand it, you understand a synapse quite well, and you'll really do well when you do certain drugs. Now, a synapse, I like to basically say it's nothing more complicated than when two cells come together to communicate. Now, they talk, and the language they're going to use is going to be neurotransmitters. Now, the synaptic terminal is part of that synapse. Remember, there's a presynaptic cell. That's the cell that comes first. Now, it can be a special receptor, but it's usually a neuron. Now, usually a special receptor is a specialized neuron. So I can say it's going to be some kind of neuron. Okay. Postsynaptic cell is going to receive commands from the neuron. It's the receiving cell. It's called postsynaptic. And there's a space, a slight gap between these two right here. We could see this slight gap called the synaptic cleft. Now, the language of a neuron is basically neurotransmitters. That's what they use to communicate. It's a chemical messenger, kind of like hormones for glands. Neurotransmitters are for neurons. Okay, they carry signals from cell to cell. They kind of, what I like to describe it is it's like a neuron passing a baton to another, like one runner passing the baton to another runner in a relay race in track. That's what a neurotransmitter does. Is it one neuron releases a neurotransmitter to another cell, and that transmits the signal to the next one. Just like how now they're stored in vesicles, they're released via exocytosis when an accidental arrives, just like we saw with the NMJ. Now there's a presynaptic membrane right here, and the presynaptic membrane is the membrane in which the vesicles that have the neurotransmitters in it, for example acetylcholine, have to pass through. The postsynaptic membrane is on the receiving cell, and it holds the receptors. The good example of that was motor end plate of the NMJ. Now remember, immediately after this happens, pretty much immediately after a neurotransmitter is released, you're going to have the degradation and breaking down of neurotransmitters using enzymes. For example, acetylcholine was broken down by acetylcholine esterase. Now there's others like your biogenic amines that we learn about. They're broken down by monoamine oxidase. There's carboxylases. There's a variety of different decarboxylases, different kinds of enzymes rather that break down different neurotransmitters. All right. Now, we can have these synapses. They can be neuromuscular junction, that is neuron to muscle. It can be a skeletal muscle, cardiac muscle, or smooth muscle. We can have a neuroglandular junction, that's neuron to a gland cell. That can be an endocrine gland or an exocrine gland. Neuron to neuron, that could be a neuron to another neuron. And those are pretty common. We'll talk about lots of neuron to neuron synapses when we do autonomic nervous system in AP2. Now, let's describe some neurons classification based on their structure. One of the things we can talk about is there are neurons called anaxonic. Anaxonic, they are having many dendrites but no axon. They're in the brain, some special sense organs, so they're kind of relatively specialized. They're bipolar cells. They have two processes. We can call those poles. One dendrite, one axon. Cell body between them. They're in the eye and sense organs. There are unipolar neurons. They have a dendrite and axon. What happens is the dendrite and axon fuse together, forming one pole. So you cannot distinguish an axon from a dendrite. The cell body is on the side, not between. They allow information to go very long distances. Multipolar neurons, which are when you usually think about neurons, you're thinking multipolar. They have two or more dendrites, using many dendrites, and one axon, so many poles. All the dendrites are a pole. The one axon is a pole. They're the most common, most numerous, most common. All your motor neurons are this. They're about as long as a unipolar is, and they carry information long distances too. So anaxonic has no axon. It's a cell body with dendrites. Bipolar, cell bodies in the middle, one dendrite, one axon on either side. the dendrite basically fuses, and you have this axon here. And then the dendrite's off here. But this kind of fuses together into one pole with the cell body off the side. And then we have our cell body with our long axon. Okay. All right. Neurons functionally can be one of three types. They can be sensory neurons, motor neurons, or interneurons. Sensory neurons we call afferent neurons because they arrive. They form the afferent division of the PNS. They're going to carry sensory information. Motor neurons, they are efferent neurons. They exit. They form the efferent division. They carry commands to the peripheral effectors, motor commands. And then there's interneurons. We call them association neurons. They're in the brain and spinal cord, and they distribute sensory information, coordinate commands. Very important in learning, like memory, planning, logic, to learn, things like that. Now, what is really cool, one of the things that blows my mind about neurons is that they all send signals the same way. They carry information the same way, yet they carry different information. And it depends on what direction the information is going and the neuron carrying it. There's some subtle differences that cause some differences that we don't get into. Our book just does not get into what makes it do this, what makes it, but we learn how it happens anyway. But they all carry the information the same way. It's pretty fascinating. Now, if we think about sensory neurons, their cell bodies are in sensory ganglia. What a ganglia is, now when we say the word ganglia in neuroscience, we mean a collection of neuron cell bodies in peripheral nervous system. When we say the word nucleus in neuroscience, we mean a collection of neuron cell bodies in central nervous system. Okay, so ganglia, nucleus, different things. Okay, now these unipolar neurons form afferent fibers, and they go from receptors to CNS. Now, there's somatic sensory that carries information about your outside world. Things like your body's position, touch, taste, smell, temperature, sound, that kind of equilibrium. Visceral sensory is internal conditions, like how acidic the stomach is, food is in my stomach, feces is in the rectum, urine is filling the bladder. Okay. Now, sensory receptors, there are three types. They're called interoceptors. Those are internal receptors. There are exteroceptors. Those are external receptors. And there's proprioceptors. These are doing body position. Interoceptors monitor the internal stuff like your digestive tract, your respiratory tract, your cardiovascular system, your urinary and your reproductive systems, okay? Now, they're going to provide things like stretch or distension, deep pressure, and pain. Exteroceptors, external, provide information about the external environment like touch, temperature, pressure, taste, smell, sight, balance, hearing, and all that good stuff. Equilibrium is another name for balance. Proprioception monitors your body's positions. It looks at the positions of skeletal muscles in your joints, like where are my legs, so that I don't have to look at my legs when I walk. When I dance, I know where my legs are when I dance. Okay. Now, motor neurons, they are efferent neurons. They form the efferent division. They carry things from CNS to peripheral effectors. The axons that leave the CNS, they're called efferent fibers. And they are going to be a big thing of the somatic motor, visceral motor, autonomic. Okay. Somatic nervous system is under conscious control, includes all your somatic motor neurons that control any skeletal muscle. They are in the CNS, their cell bodies, and they travel to the neuromuscular junction. So the cell body always resides in CNS somewhere, in a motor nucleus in the brain or spinal cord. Visceral motor, they regulate without conscious control. They control smooth muscle, cardiac muscle glands, and adipose. Their cell bodies are in the autonomic ganglia. Very important that we know the four cells, the four kinds of effectors of the visceral motor. Very important targets for drugs, okay? Now, let's talk about neuroglia. What's about the CNS? Has the ependymal cells, astrocytes, oligodendrocytes, and microglia. PNS neuroglia will be satellite cells and Schwann cells. Now, some of these do the same job in the CNS and PNS, and we'll talk about them. Now, I want you to know what each one does and where they do it, CNS or PNS. Okay? We're going to start with ependymal cells. Now, ependymal cells, they're in the CNS. They line your central canal. They line your ventricles of your brain. And what they do is they're anywhere where CSF flows. They help produce the CSF, and they have cilia on them that flow the CSF. Sorry if my dogs start to bark. It is very close to their feeding time, and I'm recording, and they're being very needy right now because they're hungry, and I'm sorry. I'm about done. Okay, the astrocytes, the neuroglia that are in the CNS, located in the CNS, they're the biggest and most numerous. I want you to think astro means stars. What do we know about stars? Stars are very big, and there's a lot of them. Okay, so astrocyte. They're the largest and most numerous. One of the things they do is maintain your blood-brain barrier. So they isolate CNS from blood. They create a framework for the CNS. So they create structure. They repair damaged neural tissue. So if neural tissue does get injured, they help repair it. They also, when they help control growth of neurons, and they help make connections of neurons. So they assist neuron development when synapses happen. They also control your interstitial fluid. That is a type of extracellular fluid around the cell, around the tissues. It regulates your carbon dioxide, nutrient, and ion levels. Another thing they do is they absorb and recycle any neurotransmitters that we don't break down. For example, if acetylcholine esterase fails to break down acetylcholine, your astrocytes can grab it up. Okay. Now, in the CNS, we have a oligodendrocyte. Oligo means few. You've probably heard the word oligarch, especially since Russia has been a big thing right now. The Russian oligarchs, the rich, wealthy Russians who, after the fall of the Soviet Union, basically owned everything. And one of those oligarchs is Vladimir Putin, and he's in charge. I mean, we probably heard the word oligarch being used in discussion with Russia. Oligo means few, dendrocyte. This is cells with few dendrites. These are going to myelinate CNS. So they make myelin sheaths. And what does myelin do? It speeds up action potentials, makes them travel faster. If the axon is myelinated, we'll call this white matter. This is part of white matter. If it lacks myelin, that's going to be in the gray matter. Now, there's a couple things. There is the myelin sheath. It looks like a little pill that wraps around your axon. Where it touches the axon is called internodes. Where it's not touching, there's a gap. That gap is called a node of Ranvier. Okay. Now, I have a picture later on that I'll show you guys. Now, microglia, they get rid of microbes. Micro means small. They are smallest and least numerous. So now, I just attended a little conference on Friday, and I saw some work that somebody was doing on microglia. And what they were doing is they were looking at how depression med fluoxetine, which is Prozac, affected a signaling system inside of microglia. And what they were trying to do is they were trying to prevent neuroinflammatory diseases. Okay, so they appear very early in development, and they remove waste and pathogens. I thought it was fascinating. I wanted to share that because I just attended it on Friday. All right. So they're your neuroglia or neuroglia. I used to always say neuroglia because in neuroscience it's all neuro. But it is actually technically properly pronounced neuroglia. And that bugs me. So, all right. Now, in the PNS, there's satellite cells and Schwann cells. Satellite cells are sometimes called amphicytes. They're kind of like the astrocytes, but in the PNS. They regulate the external environment. Schwann cells, also called neurolymphocytes, they create the myelin in the PNS. Now, one thing that I'm going to ask you on this that I love to ask on a test is what myelinates CNS, what myelinates PNS, what cells myelinate where, okay? All right, so we have CNS neuroglia, we have PNS neuroglia. Okay, so let's talk about myelin. I said I would show you guys a diagram. So it looks like little capsules wrapping around an axon here, and each of these are a myelin sheath, and these myelin sheaths wrap around it. Now, there's places where the myelin touch. These are called the internodes. They're the gaps where the myelin isn't, called the nodes or nodes of Rambier. Myelin wraps around your neuron and increases speed of action potential. It makes it faster. Okay. Now, what's going to happen is there are demyelination diseases. Two that you may run into is MS, multiple sclerosis, and Guillain-Barré. Guillain-Barré, one way that Guillain-Barré happens, I'm always hesitant to say this. I am very pro-vaccination. I'm very pro-science. There is a protein found in one of the influenza vaccines that basically is also found in the flu virus. And what would happen was it caused Guillain-Barré. It was also found on neurons. I apologize. In the flu vaccine, that protein was also found on your myelin sheaths. And what happened is we made antibodies against it, and we attacked our own, and we started demyelinating our own cells. And so sometimes there was a flu vaccine that was causing Guillain-Barré, and they had to actually go back and adjust that. They had to change it. But MS and Guillain-Barré are two demyelination diseases that you may run into. MS is a really big one, multiple sclerosis. Progressive disease usually starts out in the eyes, then starts to go to the motor. So lack of visual acuity is one of the first things, and then you'll start to get motor issues with MS, things like that. And now I'll talk a little bit about Lou Gehrig's. I should have mentioned it when I did NMJ. Lou Gehrig's attacks the presynaptic end of the presynaptic neuron. So that's why it leads to the movement disorders with Lou Gehrig's disease, alateral myeloid sclerosis, ALS, amyloid lateral sclerosis. There we go. It's late in the day. I've taught all day. It's my really busy day. All right. Now, one of the things I want to talk about is Wallerian degeneration. I don't test on the level I teach you. At best, I'm going to go ahead and tell you, at the best on the test you would ever see is the definition of Wallerian degeneration. You will see nothing more than that. But I always tell you, because the steps won't be on the exam, but it's one of the cool things about it is if you damage a neuron, it can repair the nerve a little bit using what's called Wallerian degeneration. It depends on a few things. Two big things is damage is minimal and there's good blood flow. Now, if the damage occurs, the axon degenerates. The Schwann cells will grow together. This is PNS, Schwann cells. The Schwann cells, axon will grow a bud along their Schwann cells as they'll start to bud. And those buds will grow together, and the axon will grow back together, and the Schwann cell will cover it. If you don't go further, it will not be restored if the process can go no further. But it's very difficult to do this in the CNS with brain damage. All right, we're going to stop here. This is where I want to stop. We'll pick up in the next class. Probably be Thursday before I can get the recording out. So I'll see you guys in the next one.